Okay, so welcome to explore practical first-hand experience in using SNOMED CT stream of the SNOMED CT Expo 2021. My name is Ian Green. I'm the Customer Relations Lead for Europe and also the Global Clinical Engagement Lead, and I'll be moderating today's session. Please use the live Q&A in your AirTouch device to type in your questions to the presenter, and then all questions will be answered in the conclusion of the presentation. I'm now pleased to introduce Hanne Fjeldstad Johansen from the Norwegian NRC. And she'll be presenting today a presentation entitled Modeling Genotypes and Phenotypes using GCI axioms for national sharing of emergency medical information. So Hanne, please proceed.
Thank you, Ian. So I'm going to try to go through our process of attempting to model the genotypes and phenotypes for genetic polymorphy in the SIP metabolism using the GCI axioms that are available in the SNOMED CT authoring platform. So this is just what we're going to talk about today. I'll try to give you some background information first, and then explain the problem we wanted to solve, our hypothesis and strategy, and then some discussions about what we have been doing and what we found out. And I'll be sharing some images from the diagrams through the authoring platform of what we actually made, our suggestions, and then we'll conclude. So from the start, we wanted to work with, or we are working with, the Norwegian National Health Network to connect concepts in SNOMED CT with a selection of national code systems that are created for use in a web-based solution called Kjernejournal in Norwegian, which is a national summary care record, quite similar to the international personal health record that the EU is developing. So the summary care record was developed to bridge the gap between different electronic health records and levels of care in Norway. And its purpose is to provide quick access to important health information about patients, regardless of where they receive their treatment. Oops, sorry. In this system, alert information is categorized as critical, and critical information in this context is defined as information of special importance to healthcare personnel and information that may affect treatment decisions, especially in an emergency situation. And within this category, there's been created a list of conditions that are considered to always be of critical importance, and this is called the absolute list. And among the conditions on this list is a group of conditions that are related to genetic polymorphisms that cause disturbances in the metabolism of cytochrome P450, specifically in the families 2C19, 2C9, and 2D6. This is just the user interface for Kjernejournal. This is how it looks for the user, and we've tried to put a mark around the actual registration of a SIP metabolism error. So this is our specific use case. The main goal was to represent specific genotypes using the allele star nomenclature. And an additional goal was to try to connect these genotypes to the phenotypes that they often lead to without explicitly stating the relationship with an ISA relationship. So just shortly about genotypes and phenotypes. In a broad sense, the term genotype describes an organism's complete set of genes, but in a narrow sense, the term can also be used to refer to the alleles or variant forms of a gene that are carried by an organism. And humans have two alleles at each genetic position or locus, with each allele inherited from each parent. And each pair of alleles represents the genotype of a specific gene. And the term phenotype is defined as the observable and measurable characteristics of an organism as a result of the interaction of the genes and the genotypes, as well as environmental factors and random variation. And examples of phenotypes might be eye color, blood type, or as in our case, this rate of specific enzyme metabolism. The cytochrome P450 metabolism, also called the CYP metabolism, is a superfamily of enzymes that are important for the clearance of various compounds, as well as for hormone synthesis and breakdown. And CYPs are the major enzymes involved in drug metabolism, accounting for up to 90% of the total metabolism of the most commonly prescribed drugs. And this metabolization happens principally in the liver. Most drugs undergo deactivation by CYPs, but some substances are instead bioactivated to form their active compounds through the CYP metabolism. And the effects on CYP activity are a major source of adverse drug interactions, because changes in the CYP enzyme activity may affect the metabolism and clearance of various drugs. And this leads to a need for individualized dosage adjustment or selection of drugs that do not interact as much with the CYP system for patients that are affected by this kind of polymorphic condition. And so genetic mutation polymorphism is kind of two sides of the same book. A gene is said to be polymorphic if more than one allele, which was the variant that you inherit from your parent, occupies that gene's locus. In addition to having more than locus, each allele must also occur in the population at a rate of at least 1% to generally be considered polymorphic. And genetic variations that are prevalent in less than 1% of the population are referred to as mutations. So it has to do with prevalence. So on the left here, we have we have some images from an article that has tried to chart the prevalence of poor and ultra rapid metabolization of CYP enzymes in Europe. I recommend reading it. It's in our sources. So our problem was that the existing concept in SNOMED CT only specified the phenotypes. They represented phenotype concepts without any distinction for allele placement. And so we needed to make the genotypes with the star nomenclature present in the term. That was what we set out to try to do. So through discussions, we established that for this specific use case, we wanted to represent the genetic polymorphism or the genotype, also establishing a relation to the clinical manifestation of that genotype, which is i.e. the phenotype. And within the field of medical genetics, new discoveries are made practically every day. So this includes discoveries about the mechanisms and genetic polymorphisms that govern metabolization of enzymes within the body. The clinical reality may change from one day to the next as we discover previously unknown dependencies and single out genetic markers. So the genotypes may be subject to change at a very high rate. So the potential solution we wanted to test was if using general concept inclusion, or GCI, in SNOMED CT to express the phenotype could give us more rigid modeling and less management burden for the phenotypes, and allow the genotypes to be more non-rigid in their property. For our working hypothesis, we wanted to see if we could model new concepts that contain the distinguishing features between both the genotypes and phenotypes separately. And then we wanted to see if we could establish a connection between them that was not stated as a relationship using GCI axioms. And we thought that the classifier then could do the work to infer associations between them, which would hopefully lead to a necessary but not sufficient association in line with the clinical reality, as well as reducing the workload for content management. So starting with our modeling, first we made a new procedure to represent the molecular genetic test to uncover mutations or polymorphisms in the CYP metabolism. And we placed that with other similar procedures like BRCA mutation detection tests. and tried to define it as best we could with what we had available. And then we tried to model the genotypes as types of genetic polymorphisms. We decided to name them as such as genes encoding cytochrome P450 with FSN and terms. And then we included the specific family, subfamily, and allele placement stated in accordance to the star nomenclature in the naming. And for the logical definition, we stated interprets and has interpretation relationships referring to the procedures that we made, and then added a detected value for the has interpretation. And then we established that the different genotypes had a interprets relation to enzyme metabolic function that was either increased or decreased for the has interpretation attribute in that pair. Finally, we added the occurrence and the closest finding site we could get. So this is how they look. One is for an ultra rapid, one is for a poor metabolizer phenotype, as far as we know today. And then modeling the phenotypes, we established the stated logical definitions for the phenotypes, and if they were increased or decreased metabolization or enzyme metabolic function. And then we added the GCI axioms. So this is the fun part. This association, we axiom in each of the phenotypes and stated the same attribute relationships as we used in separate genotypes. So our thought was that establishing these relations would help us keep the stated logical definition in the phenotypes constant, while enabling the classifier to infer a relation from the genotype to the phenotype that we didn't explicitly state. And our theory behind approaching the modeling in this manner was that using GCI would allow us to state the clinical truth as we know today within both findings, but also lessening the burden of management for an area that is subject to rapid change. So this is how the phenotype looks in a stated view. And then when we do the magic of classification, it turns into this after classification. And then, as you can see, the GCI axiom is no longer visible to you. And when we go through the genotype, this is the stated definition, and the parent is as we put it, and then the relationship axioms that we also added are present, but when it is inferred, it gains the parent of the phenotype that we stated with the same axioms in a GCI. So what happens is that when we look at the genotype in the browser, in the inferred view, we actually are able to gather the genotypes as children and group them within all poor metabolizers in the same genotype, and then the ultra rapid is separate actually for the other specific phenotype, which is what we wanted. So we had some challenges during our work, and we still do, but the main challenge we faced was that testing this approach in the present genetics area of SNOMED CT is still quite primitive and high level. And as such, we would have to do a lot of revamping, new modeling, structural review to be able to define the concepts, the genotypes and the phenotypes sufficiently. So per now, they're all primitive. So we are also able to define them as increased and decreased, but there are levels of poor or intermediate, normal, rapid, and ultra rapid metabolizers, which that distinction is not made as of today in our example. We would have to expand on some supporting hierarchies to be able to allow that distinction, which requires a bit more work. And furthermore, the finding site might be a bit less precise than we would like it to be, because the body structure doesn't go down to the locus level, which is debatable if it's needed or not, but that's something we looked at. Furthermore, we're not certain if our specific approach will be universally justifiable for other similar clinical ideas. We think it works for this specific use case, but we can't really confirm or deny if it works for similar use cases. That would have to be explored further. Also, the concepts that were added in the July version of the international release use a different approach that is much simpler, just stating that the genotype representation with the allele placement is a child of the phenotypes that are currently present in SNOMED CT. So it will be a question of, do we use the international model, or do we try to make our own more detailed model like this? We've also discussed if the current practical use case for GCI axioms is in fact an is-a relationship. If the inferred relation is an is-a relationship or an association, and started thinking about what other relationships besides is-a have a transitivity, and how will GCI affect these relationships. Could perhaps the GCI axioms be more of a functional type of relationship than an is-a, with an axiom that is necessary but not sufficient? We also discussed the potential of this kind of approach. So what we see is using GCI axioms to establish necessary but not sufficient logical definitions could allow for inferences that are not explicitly stated, and the practical concepts could give us the possibility of stating relations that are true and yet not necessarily constant, as in the case with the phenotypes and the SNP metabolism. These are most often caused by a distinct polymorphic genotype. However, phenotype manifestation is also affected by environmental factors, as well as a whole slew of other genetic factors that impact different parts of the metabolic processes. And thus, they're not exclusively caused by the presence of a polymorphic genotype. And in cases like these, GCIs might enable us to establish this sort of stated necessary but not sufficient logical definition. The potential of usefulness in clinical decision support for these kinds of concepts specifically is greater than the sum of their parts as well, because a logical inference between genotypes and phenotype manifestation could allow for alerts of possible manifestation of a phenotype when the clinician registers a confirmed polymorphic genotype in an EHR. And we might even want to explore the possibility of producing intentional reference sets that contain the substances or the clinical drugs that are metabolized by cytochrome P450, and connecting these to the phenotype concepts in an alert system in the health record to allow for alerts when prescribing medication to patients that have a conformed polymorph genotype. And if this is done correctly, such a solution might greatly reduce the risk of human error in prescription of medicinal products and help the clinician select more personalized medication options for these patients. So in conclusion, during our work trying to model concepts to represent polymorphic genotypes of the genes that encode the SNP metabolism, we discovered that the tools to create highly sophisticated inferences using GCI and classification are present in the AL2 web ontology language that SNOMED CT concept is logically based on. However, as of today, the content related to genomics in SNOMED CT is not developed to a point where we are able to take full advantage of the possibilities provided in AL2. We will also have to consider the place of GCIs. What kind of relationships do they actually build? Finally, for our specific use case, we will have to consider the pros and cons of releasing and utilizing content that differs from the international approach. That is a simply stated relation between the phenotype and the genotype. And we will have to decide which solution will be the best for the Norwegian summary care record and for information sharing in future. So yeah, and here are our sources. Please contact us for further information, and we're looking forward to getting some exciting questions. Thank you.
Hanne, for your presentation. Can I ask that the attendees just give us a moment while we switch over from our pre-recorded content so that Hanne and her colleagues can address the questions you've been submitting live. So please note also that this session will be available on the event platform for the next 3 months. Thank you. So hi everybody, I hope you enjoyed that presentation. Just remind the attendees, if you have a question, don't raise your hand, but can you type your question in the Q&A box and we'll aim to answer them. So your presentation has generated some discussion, as you can see from the Q&A. So we'll do them in the order they're on my screen, so not necessarily in order of importance. Do you have expected any subsumptions between those concepts that are modeled by GCI axioms? And if you did, how did you handle that?
Is it supposed to say unexpected subsumptions or?
Well, I think it's supposed to be unexpected, because otherwise, if it's expected, you'd be dealing with it anyway. So I'm assuming that's unexpected.
So we tried and failed a bit. For this one, we didn't get any unexpected inferences, but something that we briefly mentioned is that for this example, we've used poor and ultra rapid, which are increased and decreased, but the staging is also includes rapid, which is still increased, but not in the same level. So we did some tests with other stagings as well, which were expected to fail, but let's call it an unexpected inference in the same group, because they couldn't differ between each other. So we would have to make some further supporting concepts to differentiate in the GCI axiom so that the classifier could infer correctly. But for this specific example, we didn't run into any unexpected inferences right now.
Okay, so moving on a bit. So although the observable entity hierarchy is currently underdefined, have you explored how you might use the observable entity model, which might better suit your needs?
You guys have any?
No. I would really like to comment on the question from Sam on that one. The thing is, this is a finding. It's not really an observable. The test leading up to the finding or the clinical evaluation would be an observable. So you would have an observable test which would infer or would lead to a clinical conclusion, being the phenotype or the genotype being findings. So as the observable might have a model that could support this, part of our argument is that a genotype isn't actually is a relationship of a phenotype. Both are clinical findings. They belong in the clinical finding hierarchy, and then we rather tie in the tests done, and the tests will be defined by the observable entity hierarchy. So we have looked at both hierarchies, but we are pursuing this along the lines that these are actual clinical findings.
Okay. So another question off the Q&A. So the other challenge that this person has found is the requirement associated with multiple alleles. Oh, hang on, the screen's jumping. Yeah, multiple alleles. So modeling all that might mean what we might or might not within the realms of EL and using the EL profile. Have you found limitations using EL and OWL, or are the limitations more restricted to SNOMED?
That's kind of what we're sniffing on at the end of our little test project here. What profile would we have to make this not necessarily the necessary but not sufficient definition, which creates an association, and to what extent is that an association and a parent relationship? I'm not sure if I grasped the question correctly. But
No, I think that's. I think you answered. I think you answered the question. So picking up on one of the points you made, Jorn, I guess this is more directed to you. There's obviously some fundamentals to do with the structure of SNOMED as what the ISA means within SNOMED and what necessarily and sufficient means in SNOMED. Do you think that we should be opening that debate and almost addressing the fundamental structure of SNOMED driven by the project that you have undertaken here? And if you did, I'm just saying I'd support you. I'm just asking the question.
I know. I won't hold you to anyway. At least for the GCI axioms, I think it's a very relevant discussion because I think they're underexplored in SNOMED. We have searched SNOMED for how often they are used, and most of the use cases we found for GCIs in SNOMED is when there are actual flaws in the anatomy, combining groups together that aren't structured properly in the hierarchy. At least that's my impression of it. I'll be careful now. But seeing what is actually a GCI axiom, because if you have the assumption that everything is a relationship, the classifier should handle them as is a relationships when using the defined axioms. So it's just a question of modeling correctly. But when you try to do something like this, when you're saying, well, you can't actually classify a genotype under a phenotype because it's not supported by any of the foundational hierarchies, it doesn't support under the model, then you're using the GCIs to include concepts from another area. You could go all the way and you could say, I'm going to include concepts from another hierarchy. What is then the relationships? So I would think it would be interesting to see if GCI axioms could be put inside another kind of relationship, saying that I'm now inferring a relationship that isn't actually an ISA, it's some sort of other kind of relationship, seeing these lead to this finding.
So I agree with you. I think part of the reason why I said I'd support you, this has come up in a number of conversations with genomics groups about how to use SNOMED, particularly when it comes to analytics and what different types of meanings, if you take that from SNOMED, what it actually means. And it obviously almost goes to the fundamental structure of SNOMED rather than particular areas of content. There's something above that in some ways. The one challenge, I'm sure you'd all support this in terms of the genomics area within SNOMED, is a push from yourselves and end users to fully define all the content within that area, which would make your job easier, I'm guessing.
Very much so.
It's certainly, I mean, one of the things that we're looking at, so one particular group that I know have been looking at it is the Australians. So they've been doing quite a lot of work around genomics procedures. And it's something which I think we should look at sharing resources in some ways and bringing in some of the work which you as members are doing. And then there may be some, I'll call it integration work at the level of the international. But I think this is about sharing some of that work, particularly with genomics and the need for accessing the expertise. So you're looking at expertise in SNOMED and genomics. It's not a huge group of people out there at the moment. So in the interest, as we're almost to time, I'd like to call this Q&A session to a close and this session to a close. I'd like to thank all our presenter and her colleagues. I'm sure it's been well received, and I've certainly enjoyed it. But thank you all again, and hopefully you'll enjoy the rest of the expo. So thank you.
Thank you.